Hi all, we have studied chapter 1, Reproduction in Organisms. We are moving on to the next chapter, chapter 2, Sexual Reproduction in Flowering Plants. By now we are clear that sexual reproduction is a better method of reproduction because it brings in variations. Last year, chapter 3, we studied Plant Kingdom. We studied various groups under plants, namely the algae, bryophytes, pteridophytes, gymnosperms and angiosperms. And you know that angiosperms are commonly known as flowering plants. Why we concentrate on sexual reproduction in flowering plants? Because they are the dominant group of plants in the present day. They are most distributed plants on the earth. So we concentrate on flowering plants. Like before, here also we concentrate on three events, namely pre-fertilization events, fertilization and post-fertilization events. To remind you, it's a part of Unit 6, Reproduction, which is for 25 marks for the final exam. Under topic sexual reproduction, let us take a recap from the last year. Last year, 5th chapter, Morphology of Flowering Plants, we studied the parts of a flower. So this is your introductory topic. 2.1, flower, a fascinating organ of angiosperms. It's a fascination. To decorate some place, you use flowers. To worship God, you use flowers. To decorate a dead body, you use flowers. We use it in several occasions. This beautiful structure comes in various colors, structure, with aroma, so much of variation. I have written a diagram of a typical flower, simple flower, to make you understand the different parts. We studied last year that flower is a modified shoot. Shoot is a stem-leaf complex. Flower is a modified shoot. The flower has a stalk. The stalk is called pedicel. The tip of the pedicel is thalamus. On the thalamus, you find 4 orals of a flower. The units are arranged in 4 layers, or say 4 nodes. And those nodes are superimposed. The outermost oral forms the calyx. It's made up of sepals. It is followed by the second oral made up of corolla, which is having units called petals. The third oral, we call it androecium. It's made up of units called stamen. The fourth and the innermost oral is the gynoecium or pistil. It is made up of units called carpels. These units, each of these units are equivalent to a leaf. This is a stalk, which is a stem. That's why we call flower a modified shoot. It's a stem-leaf complex. Stem-leaf complex. The flowers occur in clusters and you call it inflorescence. I have given you for summary the details of a flower, parts of a flower. So in this chart you can observe the parts of the flower can be classified into 2 parts, namely the accessory and the essential orals. Oral refers to the circles. The flower has 2 types of orals. Accessory orals, essential orals. Accessory is also called non-essential, something which is not very important for sexual reproduction. It is only supportive in function. It supports reproduction. It does not directly participate in reproduction. Accessory orals are 2 in a generalized flower, and they are calyx and corolla. Calyx is the outermost oral. It's made up of sepals. And you can see the sepals. In a hibiscus flower, you will see a green cup and the green cup represents calyx made up of sepals. ದಾಸವಾಳದ ಹೂವಿನಲ್ಲಿ ಒಂದು ಪುಷ್ಪ ಪಾತ್ರೆ ಗ್ರೀನ್ ಕಲರ್ ಕಪ್ ಇರುತ್ತೆ, ಅದನ್ನು ಕೇಲಿಕ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಅದರಲ್ಲಿರೋ ಯೂನಿಟ್ಗಳಿಗೆ ಸೆಪಲ್ ಅಂತ ಕರಿತೀವಿ. Above that is corolla. Corolla is brightly colored and it is variously colored. And corolla is made up of petals. It can be various in color. So these are the petals. The number is also variable. Think of a rose flower. You see hundreds of petals. Whereas a flower like Vinca rosea, there are only 5 petals. These are the topics you studied last year. So you understand that petals are attractive in function. They attract the pollinating agents. Accessory orals are followed by essential orals. The essential orals are 2, andrecium and gynoecium. Andrecium is made up of units called stamen. Stamen is technically a leaf. Stamen is made up of sterile part called filament. So why you call it sterile? Because it does not produce the reproductive cells called pollen grains. So this is sterile part and this is the fertile part, the most important part. The filament holds the fertile part up in the air so that pollen grains can be easily distributed. Filament ends in anthers. Generally there will be 2 anthers. So you call this stamen a dithecous stamen, where di means 2. Anthers break open and release dust-like particles, you call them pollen grains. And these pollen grains represent the gametophyte stage of the life cycle. Remember, last year we studied 2 generations in each plant group. Every plant has 2 stages, sporophyte and gametophyte. This whole structure what you see, the flower on the plant, the root stem leaf, the whole plant, think of any plant of angiosperm type. All the plants, whether it's a coriander plant or a jackfruit tree, a coconut tree or a curry leaf, they all represent sporophyte stage. They reproduce by producing spores. And that sporophyte gives rise to gametophyte. These dots represent gametophyte stage. We call it pollen grain. So you see the alternation of generation from the sporophyte stage to gametophyte stage. The flower reproduces by producing spores. The spores give rise to gametophyte. This is called male gametophyte, the pollen grains. So what is the function of sepal? Protection. What is the function of petal? Attraction. What is the function of stamen? Produce pollen grains. The innermost oral, the 4th oral I am now concentrating on is pistil, the female part of the flower. Pistil, also called gynoecium, the most important part, because this gives rise to fruit. Gynoecium is made up of units called carpels. So this is gynoecium made up of carpels. When I say calyx, I could show number of sepals. When I say corolla, I can show number of petals. When I say andrecium, I can show number of stamen. But when I say gynoecium, usually it is in one bundle. The reason is the carpels are united. They form one structure. The condition is called syncarpous condition. Then how do I know that it is made up of many carpels? I said the gynoecium later becomes a fruit. So when I cut a fruit, depending on the number of chambers I come across in the fruit, I guess that it is made up of so many carpels. For example, if I cut a fruit of lady's finger, bendekai, when you cut it, you see 5 chambers, indicating that the fruit of lady's finger is made up of 5 carpels. Well, of course, sometimes it can be 6 also. If I cut a fruit of cucumber, sautekai, you observe that there are 3 margins at which seeds are attached. This indicates that cucumber gynoecium is made up of 3 carpels. On a coconut shell, you find 3 holes. This indicates that coconut is made up of 3 carpels and so on. So this number is variable, 1 to many number of carpels. In an orange, when you open the skin, you see number of compartments, something like 8 to 10 compartments. That means they are made up of 8 to 10 carpels. So carpels generally form one bundle. That's why you cannot individually show them. It's seen a single bundle. And this gynoecium has 3 parts. The basal part called ovary enclose ovule. The ovule is attached to a tissue called placenta. We use the same word even for a human being. The connection between the mother and the baby is also called placenta. We use the same word here also. Plants also we use the same word, placenta. So this basal part represents placenta, to which the ovule is attached. It takes food from the plant to grow into a seed. Ovule is the future seed. Ovary is the future fruit. To make this ovule convert into seed, it has to be fertilized because it contains egg, the female gamete, and the male gamete is within this pollen. The sperm is within the pollen. When the sperm meets the egg, the act of fertilization takes place, and this needs pollination. Last class we were discussing that the gametes are transferred through various methods. For lower organisms, the gametes themselves are motile. For flowering plants, it's pollination. In case of animals, they come together. So we need a process now called pollination. So these pollen grains are made to land on the tip of the gynoecium, and the act is called pollination. This is achieved with the help of wind, or some animals may help it, a human may help it. Ultimately, they land here and they produce a tube. The tube carries the sperm. Sperm finally meets the egg and fertilization act is achieved. And once it is achieved, this structure, the ovule, develops into a seed, the ovule develops into a fruit, and this style and stigma, they degenerate. To sum up the function of different parts of gynoecium, now we have understood stigma receives pollen grains. It has to check whether it is the right pollen grain or not. If it is the right pollen grain, it is allowed to grow through. Style holds the stigma up in the air so that it will receive maximum pollen grains, so it can make a choice, the best out of the available. So style, it has a function of conducting the sperm from the stigma towards the ovary. Ovary forms the future fruit, ovule forms the future seed, and within the seed lies the embryo, the future generation. So this is a LS, longitudinal section of the flower. The same thing, now I will sum it up here. Pistil or gynoecium is made up of units called carpels. Generally carpels cannot be counted by seeing the gynoecium because they are united. And the carpels have 3 parts: the basal ovary, middle style, terminal stigma. And the ovary enclose ovules, and the ovule has embryo sac. So this white structure I have written here represents embryo sac, and that embryo sac encloses the egg. From the male side, sperm is carried and the female carries the egg. When they unite, you call this process fertilization, and the product is called zygote. The zygote is now subjected to 2 events. Remember, last class discussed cell division, cell differentiation. The zygote has to divide so that the number is increased. Then they have to specialize. So finally, it results in an embryo. So all these events, they constitute pre-fertilization events until the development of sperm. Union of male and female gamete constitute the second event, fertilization. It is followed by post-fertilization events. So this chapter is all about understanding the 3 events: pre-fertilization event, fertilization, and post-fertilization events. Till now, we have studied the parts of a flower. We understood that there are 4 orals. They can be distinguished into 2 types: accessory and essential. Essential orals are orals that are necessary for production of seed. They are 2: androecium and gynoecium. Our emphasis in this chapter is to understand various microscopic details about androecium and gynoecium, understand stepwise how exactly sexual reproduction takes place. Let us now concentrate on the second topic of this chapter: pre-fertilization structures and events. Obviously, we need to concentrate on 2 different structures, namely the male part of the flower and the female part of the flower. So on the board I have now put the diagram related to male part of the flower. Of course, the colors are imaginary because only the yellow color is predominant. You don't see these colors when you see in a microscope. It is just to distinguish different cell layers. I have written different colors. A stamen is technically a leaf. Phyllos is leaf. It's called microsporophyll because it produces microspores. So the name microsporophyll. A stamen has a filament. This can be equated with the petiole of a leaf. There are 2 anther lobes. They can be equated with the lamina part of the leaf. The midrib of the leaf can be compared with the connective, a tissue that carries nutrients towards anther lobes. Of course, the leaf tip part is missing here. You don't find an extension above the anther lobes. So technically, stamen is a leaf. It produces pollen grains. When the stamen produces pollen grains and the pollen grains get ready to get dispersed, there will be a weak zone in the anther. This dotted line represents that, and those weak zones break open and release the pollen grains. I have enlarged this to show you how it breaks. So this dotted line, I have shown it here. When it matures, it breaks and releases the pollen grains. So these pollen grains, either with the help of insects or with the help of wind, they travel in the air. It's a very random movement, and finally they land on some region. So if it is a stigma, and if the stigma of the same species flower, then there is a chance of fertilization. So it's totally random. We just don't know where exactly the pollen grain will land on. So these pollen grains are produced inside pouches. You can see there are 4 pouches. So these pouches can be more clearly represented when I take a section of a young anther. So I take this anther, cut it. When I take a transverse section and observe the cut end, I observe 4 sac-like structures, 4 bag-like structures. Each of these bags, we call it microsporangium. In a dithecous anther, there will be 4 microsporangia. The microsporangium is protected by wall layers. Each of those wall layers have a specific function. Have you got this diagram? You understood this diagram? These are anther lobes. Anther lobe number 1, anther lobe number 2, 1 and 2. And this connective tissue is seen here. Each anther lobe, when you cut it, you are able to see 2 microsporangia. So in total there are 4 microsporangia. Now let us enlarge 1 microsporangium and get more details about it. So this is a diagram you have to practice for the exam. So this is a question you expect in the exam. Explain the structure of mature anther. When anther is fully grown and it is ready to burst out, what are the cells you come across? So this is the diagram you will write. I have enlarged a part of this anther. I have given you the details of various types of cells. The outermost layer is epidermis. This is quite obvious. You have studied the same term with respect to every other plant part. In case of root, stem, leaf, we use the same term. Of course, in the root we call it epiblema. So this is a structure protective in function. Epidermis is the outermost layer made up of barrel-shaped cells closely arranged without intercellular spaces. The function is protection. It is followed by a layer of large cells. Cells are quite large in nature. Inside the cell, we find some spindle-shaped structures, some spring-like material. They are arranged in a radial manner. The orientation is in a specific direction. These are hygroscopic in nature. The meaning is they absorb water and expand. They have the ability to expand. Like a rice grain, for example, when cooked, it expands. A similar nature you notice in this. When they absorb moisture, they expand. Thus, they help to break open the anther wall. So the anther breaks here. It breaks here when the endothelial cells expand. When these cells expand, the anther breaks and thus pollen grains are set free. There is one more cell layer. This can be one layer, sometimes two layers. We call it by the name middle layer. Because it does not show any speciality, we assume that it has no specific function. Middle layer, it is simply a cushion-like cell layer in the anther. It is followed by innermost cell layer. We call it tapetum. This tapetum has multinucleate cells. You can guess why multinucleate. The function of a nucleus is to produce RNA. RNA produces proteins. Nucleus has some X amount of DNA. If there is one nucleus, it would result in X amount of protein. So if there are two nuclei, it should be able to produce 2x amount of proteins. If there are 4 nuclei, it should be able to produce 4x amount of proteins. That is a simple logic. Why your mother prefers a gas stove with more burners than a single burner? With more burners, she can cook quickly, she can finish her job quickly. Similarly, the cells with many nuclei are able to finish their job quickly. They can provide food to the developing microspore mother cells quickly. That's why many nuclei, multinucleate condition we call it. This is due to a special mechanism called endomitosis, where there is a nuclear division not followed by cytoplasmic division. Each cell will possess many nuclei. You cannot have too many cells because there is no space. So the best idea is each cell is given additional responsibility. Having many nuclei, it becomes highly synthetic. Tapetum secrete materials for growth of those future pollen grains. We call them by the name microspore mother cells. The microspore mother cells in future gives rise to pollen grains, providing nutrition and later providing some enzymes to separate the cells, providing some material called sporopollenin. So various functions are performed by tapetal cells. Epidermis, endothelium, middle layer, tapetum. And tapetum enclose microspore mother cells. In the exam, when you answer a long answer question, you will draw this diagram and explain these cell layers and mention about microspore mother cells, and they later give rise to pollen grains. That is the content you are supposed to write for the long answer question. So we studied the structure of mature anther and understood that stamen, a modified leaf, at its free end produce pollen grains, and pouches in which pollen grains are produced is called microsporangium. It is protected by wall layers namely epidermis, endothelium, middle layer and tapetum, and within the tapetum exist microspore mother cells. In this chart, I am continuing the topic. The first short answer question topic is Microsporogenesis. Microsporogenesis. Genesis refers to production. Production of what? Production of microspores. Production of microspores from microspore mother cells. In the previous diagram, you observed that the anther encloses number of cells, and each cell I labeled it as microspore mother cell. So let us take a link from that and understand one cell, what happens to it during microsporogenesis. Till now, it was a diploid cell. The whole of the structures you saw before, the root, stem, leaf, inflorescence, different parts of the flower, they were all diploid. Similarly, this cell is diploid, means it has two sets of chromosomes. Till this stage, it represents sporophyte generation of the plant body. Now, it is entering into the gametophyte stage because it is now undergoing meiosis, the reduction division. Microspore mother cell, there are hundreds in each of the pouches. They undergo meiosis, the reduction division, and produce 4 cells. Last year, chapter 10, under the topic cell division, we studied about meiosis. We understood that in meiosis, the number of daughter cells produced will be 4. So this bunch of 4 cells, we call it by the name tetrad. Tetra means 4. Well, you are able to see 3 cells only in this diagram because the 4th cell will be behind. It's a cluster of 4 cells. In any one given view, you can see 3 cells. So there are 4 cells making a tetrad. They are haploid. They are a product of reduction division, and this process is called microsporogenesis. So the question appears as, write a note on microsporogenesis. The answer is formation of microspores from microspore mother cell through reduction division is microsporogenesis. Microsporangium encloses hundreds of microspore mother cells. Upon meiosis, each microspore mother cell gives rise to a group of 4 cells. It is called tetrad of microspores. They are haploid in nature. And you write this simple diagram. Now, these 4 cells are cemented to each other. There is a material called callose binding those 4 cells. They need to separate and move away independently. To separate them, the tapetal layer, the innermost layer of the cells in mature anther, secrete a material called callase. So this is callase enzyme. It ends with ASE, that is a suffix. Callose is a binding material. It can be digested by enzyme callase. When tapetum secrete callase, the callose material is dissolved and the cells separate, and you get 4 microspores. So like this, if there are some 100 microspore mother cells, it would result in 400 microspores. So 4 times the number of mother cells, the microspores are produced. This is one short note question. We are moving from outermost to innermost. We are going further details. Now this pre-fertilization event concludes with formation of sperms. Let me consider one microspore and show you what happens to this microspore later. I put the heading development of pollen grain or male gametophyte. You remember angiosperms have two generations, sporophytic generation, gametophytic generation. Sporophytic generation is dominant, gametophytic generation is reduced to few cells. From sporophyte to gametophyte, we are understanding the concept. So this microspore now has undergone a little change. There I showed just one membrane, but here I have shown two membranes. The microspore is now loaded with some additional material by the tapetum, and I have written it in yellow color. So this line represents an additional cell wall. Already there is a cell wall written in white color. It's called intine. On this, a new cell wall material is laid. We call it exine. And you can observe that intine is continuous, whereas exine is deposited in irregular pattern. Some regions exine is not deposited. Exine is made up of a material called sporopollenin. This sporopollenin is chemically resistant. No chemical on the earth can destroy sporopollenin. So this layer should not be present in some regions so that the sperms from inside can come outside. The sperms have to travel out of this structure. That's why there are some regions left not deposited sporopollenin, and we call those gaps the germ pores. Germ pores. So this is the starting material for our understanding of development of pollen grain. It's a haploid cell called microspore. It has two wall layers, inner continuous intine, outer discontinuous exine. Exine leaves behind gaps called germ pores. This is an imaginary diagram. I have taken a sectional view of a microspore. This cell now undergoes mitosis. You have studied that meiosis cannot repeat. Meiosis happens only once because the chromosome number is reduced from diploid to haploid. You cannot expect one more meiosis. Then it would become n by 2, which is meaningless. So now it can only undergo mitosis. Mitosis is somatic cell division where the chromosome number is maintained the same division after division. So there is a mitotic cell division resulting in two-celled pollen grain. You notice one interesting thing here, that even though the nuclear divisions are equal, the cytoplasmic division is not equal. One cell produces small, we call it a lenticular cell, the other cell is large. So there is a difference in the volume of the cells. The small cell, we call it generative cell. The lenticular cell is also called generative cell. Another cell is tube cell or vegetative cell. This cell will produce the tube, the pollen tube in future. When we study the topic fertilization, we will discuss about that. The generative cell now undergoes mitosis. It will give rise to two male gametes. We call them sperms. These sperms are very special. You don't find any tail for those sperms. They are not having any tail. They are just some rounded off cells. So you can see two mitoses resulting in three cells. Usually our mind goes like this. So first mitosis, one cell divides from two cells. Second mitosis, two cells should produce four cells. But how three cells? It's because when first mitosis takes place, two cells are produced, but out of two cells only one cell will undergo one more mitosis. That's why it is not four but only three cells. The larger cell, already I told you, it's called tube cell or vegetative cell. The smaller cell, the generative cell, after division we call them sperms, the male gametes. So even the flowers produce sperms. These stages happen one after the other. At what stage the pollen grains are released by the anther. In case of dicots, usually the pollen grains are released at 2-celled stage. And in monocots, they are released at 3-celled stage. The pollen grains carry these sperms to the stigma surface. We call this process pollination. It is then followed by fertilization. How long they stay? Their survival is only for a few hours. Once after the release, they die if they don't find a suitable substrate. Interestingly, some examples like cycads, one of the gymnosperms, they may stay for several months. It's a very special case. And I told you this is all sectional view. If you, instead of seeing a sectional view, see the entire pollen grain, it may look like this, with so many spines all over, and different species produce different types of pollen grains. They are not always the same, they are different. So if you are a specialist, if you are a person specialized in studying pollen grains, you must be called a palynologist. The topic is palynology, study of pollen grains. You will be able to identify the plant species just by seeing the pollen grain. The pollen morphology tells you from which plant the pollen grain is derived from. So in a city area, you see a lot of weeds growing all over, for example Parthenium. And if you collect pollen grains from the atmosphere and examine the microscope, you will find a lot of Parthenium pollen grains. Why city people suffer from allergic reaction, asthma-like symptoms? It's because of the allergic reaction between the body and the pollen grain. So the pollen grains land in the nasal cavity, our body reacts to it, resulting in respiratory trouble. The pollen grains can be condensed, they can be made into capsules or say syrup, a tonic. The pollen grains can be suspended in sugary syrup and you drink out of it. These are used by athletes, they are given to race horses to improve the stamina. Pollen grains can be frozen and stored in liquid nitrogen for a later use. You can simply freeze this, they are very small, dust-like. In a small bottle you can hold millions of pollen grains. They can be just packed and frozen in liquid nitrogen so that you can use it later. You can store it for a later use. We call this process cryopreservation. Cryopreservation. So these are some of the utilities of pollen grains. It is also used in checking the quality of the honey, like what type of pollen grains are seen in honey. When you identify that, you understand how good the honey is, the quality of the honey. They are used in forensic research and so on. So various such applications. So this completes the male side of the story, the pre-fertilization event in the stamen. Now we need to study the female side of the story. What happens within the gynoecium? Once we understand both, then we have to understand gamete transfer. Now we have understood gametogenesis. Later there will be gamete transfer. It is followed by fertilization. And then comes post-fertilization events.